Zlatko, please.
Thank you very much. I hope you can hear me well.
Yes,
Um,
we can hear you very well.
Very good, excellent. I will talk about a topic that is of great interest to us physicists. I've always been interested in quantum biology, but again, I think this is mainly from the physics perspective. I simply I don't know enough about biology to really know whether these things are relevant. But I think it's a fascinating question for physicists, chemists, and biologists equally from different perspectives. So what I wanted to tell you about is um, an experiment that we thought about a couple of years ago, which I think is a very simple and cute one. And it targets this question. Let me see if you can see the next slide. I'm going to try to change the slide. That's it. It targets this question of macroscopic quantumness. So the big, as you know, the big question in physics is whether there is a limit um, to in terms of maybe the size or the complexity of the object, whether there is a limit to the superposition principle. So can we really interfere in the quantum sense anything that we want? So this is this first question that drives most of our current research, and it impinges on many different areas in physics, on our technologies, as you know, as well. And of course, the answer is that we don't know. And that's why it's exciting to probe this limit and see whether quantum mechanics would collapse. And then, of course, the next question is um, if there was a limit to the validity of quantum mechanics, why would that be? And actually, again, we don't really have any, we have some arguments about some absolute limits where we simply run out of space or time or energy or whatever other resource. But we don't know if anything prevents quantum effects. Gravity, of course, is an obvious candidate to do something interesting, maybe. And there are many speculations, as you know, that gravity might fundamentally limit quantum mechanics. But again, we don't know. And I think experimentally we're getting closer and closer to that domain, which has always been interesting to a physicist like myself. And then, of course, what's relevant to our meeting here is that sometimes life is taken as playing possibly the role of the ultimate observer and maybe collapsing the quantum wave function. And it's along these lines that I want to tell you about what we thought would be cute to do. And then there are obviously, I think we've only made the first steps. There are many possible improvements that we can make along these lines, and I will discuss them in about 10-15 minutes time. So the next slide is just as a motivational quote. I think Niels Bohr gave a lecture as early as 1933 entitled Light and Life. You can find actually this in various collections of his writing. And I think Bohr was one of those people who saw complementarity more or less everywhere. So, you know, if I read out this quote, he basically saw an analogy between physicists' attempts to characterize the atom and biologists' attempt to characterize the cell. And then basically, of course, living cells are made of ordinary matter. And therefore, you know, should conform to chemical analysis. But the matter was organized in a complex and particular way. So then Bohr would go on to say, to study the chemistry, the organization had to be destroyed. While to study organization, one has to operate at the level at which chemistry is invisible. So then, you know, Bohr was one of those people who kind of pushed this idea of complementarity from the quantum realm almost into anything else that he could think about. And then he proposed this kind of complementarity that, you know, either you will be able to discover the chemical in this, for our context, quantum mechanical features of an organism, or you'll be able to understand its organizational hierarchy, but you won't be able to do both of them at the same time. And he concludes, just as velocity and location are complementary properties of an electron. So he had these very interesting speculations that somehow, you won't be able to investigate a living organism and at the same time show its chemistry or quantum physics and exhibit these properties simultaneously. And this is a very interesting statement, and most likely untrue, I would say. And in fact, in what follows, I would like to tell you that you can do certain things to basically have a living organism and at the same time superposed or entangled or whatever else you want to do. So this brings me straight to the experiment. It's a very simple slide. I'll show you a few pictures out of this experiment. So at some point, I was very lucky to meet a person called James Martin, who gave a very big donation to Oxford and set up the Oxford Martin School. He approached me and he gave me something like £2 million over a certain period of time to play around with these quantum mechanical effects and possibly in living systems and try to reverse engineer them and to see whether we can learn anything for our own technologies. And one of these experiments is the one that I'm telling you about, which I think the idea started maybe 2 or 3 years ago. The experiment was completed at the end of 2016, I think, and it was published last year as well. So you can read more about it, but it's a very simple experiment where you take, I think the idea was to move away from doing bulk experiments. Most of the experiments, as you know, in quantum biology are really bulk experiments. And the bulk experiments can frequently hide, obviously, quantum effects simply because of the various averagings and so on. In some sense, even what I am now presenting is bulk. But as far as the bacteria are concerned, they are really at the level of single bacterium that's coupled to the light field. So basically what a colleague of mine, David Coles, who was the lead on these experiments, he was at Oxford then, now he's moved to Sheffield, has a job in Sheffield. But basically what he did is he made a microcavity into which he injected these green sulfur bacteria. These are the ones that, I will show you an energy diagram of that. These are the ones that photosynthesize, and they have this FMO complex that's extensively studied in the field. But they've actually managed to inject a few bacteria into the cavity. I think under the focal spot was basically one to two bacteria, really on average. Then he shone white light, and I think this is going to be important from our perspective. This was completely incoherent radiation, not a laser light. It's just like a lamp. He shone that through the cavity. And it was just a pump and probe, basically. The bacteria got excited. Then after some time, the cavity leaked radiation, and you could study the spectral properties of the emitted light. And what was interesting again is that he could, during his experiments, confirm that bacteria are both entangled to the light field, to the photons inside the cavity, and at the same time they are perfectly alive. And I will tell you a little bit how we thought about it, how we could confirm both of these things. The next slide just shows you some nice images under the optical and electron tunneling microscopes of these bacteria. So basically the inset A and C are just the bacteria. In the inset C, you can actually see green bacteria on the right hand side, and you can see a bacterium that's absorbed the dye. After which it's no longer living, basically, but then you can observe this dye radiating inside the bacterium. So that was a kind of a very simple witness of whether this bacterium was alive or dead. And you could, in the insert B, you could see basically the spectrally resolved emissions from the bacterium, the green light, and the blue one from the bacterium once this dye molecule has actually penetrated. And so the experiment is very simple. Let me show you the energy diagram of the bacteria. Again, extremely oversimplified. Whenever a physicist looks at these complex molecules, we get a headache because they look very haphazard, very asymmetric. They are very difficult to treat in the usual way that we treat organized and symmetric solid state systems frequently. So basically, at the top of it, you see the smallest wavelength, if you like, the largest energy at which the bacterium absorbs 750 nanometers. So this is basically this famous antenna where the photons get absorbed, and then they slide down this energy scale, down basically to the base plate, then to this FMO complex, where people have, of course, as you know, analyzed this and studied this experimentally extensively on its own, taken out of this bacterial context. And then ultimately this excitation slides down to the reaction center, where it gets converted into energy and the ATP cycle and so on. What was relevant to us in this experiment is simply this first resonance at 750 nanometers where light is absorbed. And that's actually the transition that was modified because of the bacterial, of the light presence inside the cavity. So basically, the experiment found that it was impossible to regard the bacterium and light as two separate, disentangled, if you like, in our language, entities, but you had to really think of them as very strongly coupled to one another. So what the experiment really achieves is a very strong coupling regime between the optical field in the cavity and the living bacterium. And that was very, very exciting to us. So if I go on to the next slide, that's the one. You now see the images from the experiment. And what happens, I will show you the model very, very briefly. What happens is that the energy level that's relevant here actually undergoes a splitting that in the quantum optics jargon is known as the vacuum Rabi splitting. So what happens in these images, you can see two energy levels that normally would be degenerate if there was no coupling between the light and the bacterium. So think about the bacterium being excited and the cavity having no photons, and then the bacterium being de-excited and the cavity having one photon. If the two systems were really independent, if they were not interacting, then these two energy levels would be degenerate. They would have exactly the same frequency, if you like. But because of these strong couplings, these two energy levels actually are resolved, and you can see them separately. And if you look at the x-axis, as you vary the length of the cavity, which means as you change the modes, as you change the frequencies that can exist, the photon frequencies that can exist inside this cavity, you can actually look at different energy levels. In this case, Q equals 2, then Q equals 3, Q equals 4. I think the top images are just sliced through the bottom images, if you like, which is the full picture of this vacuum Rabi split. And if you're interested about more of this stuff that was observed, you can actually read the paper that's quoted down there, which is the one that presents the experiments. And in this case, you could see this vacuum Rabi splitting between different levels. And I've got these three different frequencies that are quoted there, from which you can even actually estimate the strength of the coupling between the bacterium and the cavity field. And now just a very brief summary of how you would model this as a quantum physicist. This is really the simplest model we could come up with. And again, there is a recent paper that I cite down there where we published this theoretical analysis. But the analysis is simple to the degree that you take a Hamiltonian where you assume that both the light inside and the bacterium can be described as a harmonic oscillator. I mean, this is a huge oversimplification. Of course, the light field is just a bunch of harmonic oscillators. But when it comes to the bacterium, what you should think about is that it consists of millions of electric dipoles, each of which you can think of as a two-level quantum system. So you can think of this dipole either being de-excited in the ground state or excited, having one exciton, if you like. And if you think collectively of all of these dipoles, millions of these dipoles present inside the bacterium, you can ultimately map all of the two-level quantum systems, all of these million qubits, onto effectively a harmonic oscillator, which I would call the mode B in this Hamiltonian. So you've got the mode A, which is the light field. You've got the mode B, which is simply the collective excitations of all the dipoles inside the bacteria. And then you've got this coupling governed by this strength lambda, which tells you actually how strong the coupling is. And I think it's enough to simply assume some kind of XX coupling between the two. So in the second quantized notation, this would be A plus A dagger of the light field coupled to B plus B dagger of the bacteria. And the thing to note is that this lambda, the strength of coupling, of course, depends on the dipole strength D, it depends on the electric field strength E, and it depends on the square root of the number of dipoles that exist. And actually, because we know the D and the E quite well, and lambda comes out of these experiments, I think it's called omega on the previous slide, you can actually estimate the number of the effective dipoles, this n, square root of n comes out of the experiment. And the punchline is that actually what the experiment managed to show is that you get this entangled, highly entangled state between the light, and this is this psi g that I presented, and the cavity. So basically, you either have n photons in the cavity, and you have capital N minus n excitations in these bacteria, and then they're all superposed with certain amplitudes Cn. And in this very simple model, you can actually do the full analytical treatment of all of this, and it comes out very nicely and matches the experiments. So here is this picture that you can actually see these. You can see these living bacteria. And the reason why we know they are alive is because there is this dye molecule that's also present in the bacteria. And in fact, the dye molecule is not inside the bacteria during this experiment. So you can observe both the vacuum Rabi splitting and the green light shining out of the experiment at the same time, which is actually our evidence that they're alive and strongly on resonance at the same time. It's very interesting. So that's basically the picture B, showing you the green emission light and that the bacteria are strongly coupled and alive. And what's interesting to people like me, again, coming from quantum physics, is that you can even actually analyze the amount of entanglement between these dipoles inside the bacterium and the cavity field. And as you change the size of the cavity, you can actually tune this coupling. And this picture here simply shows you that at some point there is, as it were, a phase transition when the frequency lambda c of the coupling matches more or less half of the resonant transition of these dipoles. And at that point, you have a maximum amount of entanglement between the bacteria and the light. And in fact, you can even calculate in the experiments. I think if you normalize entanglement so that when the state is maximally entangled, you get 1 unit of entanglement, then in these experiments, we've had a bit under maybe 20% entanglement in the whole system at some point, which was very exciting to us. So bacteria were really entangled to light as far as we are concerned, and at the same time, they are alive. You can actually see that they are alive. Now, I want to say a few more things about this because the experiments are extremely basic. I think this is towards probably the end of my presentation now. But if you say, is there a way in which you could actually improve these experiments and test them a bit more, the quantum effects, then the first thing that comes to mind is that maybe you can induce Rabi flopping between the two vacuum Rabi split level. So basically, not only can you now spectroscopically observe this splitting, but you can maybe drive coherent transitions between the two energy levels. So you would really take these two levels and treat them as a qubit and show that you can actually make any coherent superposition of these states. I think this would push the quantumness one level up. And then, of course, if you want to be even more precise and pedantic in terms of quantumness, you'd be trying probably to do some kind of entanglement tests. So you would really like to measure observables individually on the bacteria and separately on the light field, where you could maybe measure complementary observables and show that their correlations go beyond anything that can be explained classically. Having said all of these things, these are things we have in mind, and I'll show you some pictures what we want to do. I'm citing at the bottom of this slide a classical explanation of the vacuum Rabi splitting. So I think that's why you have to be very cautious when you're claiming quantum effects, because if you take two classical harmonic oscillators and you couple them and you drive one of them, very much like you would drive dipoles in this case with the external white light, you could actually get something that's pretty much analogous to the vacuum Rabi splitting. So one of the main issues we've had with our referees when we tried to convince them that this was quantum was simply to go beyond this classical model and argue that because light we know is quantized, and of course we have ample evidence from many other experiments, therefore we must conclude that the bacteria themselves are also a collection of quantum dipoles, and they really need to be treated as quantum systems. What doesn't work in this context, and that's very interesting, is the semi-classical model. So actually, if you only quantize light, but you don't quantize the bacteria, you will actually not be able to observe any vacuum Rabi splitting. So either you should think of both of them as classical, as it were, or you should think of them both as being quantum. But the intermediate regime is really insufficient. And of course, because light is quantized, like I said, we know that the bacteria should be quantized as well. Now, the interesting thing maybe from the biological perspective, and we've discussed this a lot as well, Dave and myself, is can we make the bacteria more efficient at photosynthesis through this strong coupling? So could you imagine that when bacteria is inside this microcavity, you can somehow tune this interaction to the extent that the bacterium actually is improved? And of course, there is the other regime where you take this energy level so far out of resonance that actually you make the bacterium less efficient. And the wildest kind of idea to a quantum physicist is always, can we somehow make a coherent superposition of these two things? So we thought, can we think of an experiment where we've simultaneously shifted the bacterium into the regime of being less efficient and into a regime of being more efficient. So you kind of get into this dead and alive Schrödinger cat state. And I think this would be hugely interesting from the fundamental perspective as well. I'm not sure if this would have any biological implications whatsoever, but it would be certainly an experiment that would be fun to do. Anyhow, finishing with this talk and going in the entanglement direction, what we are trying to do now, and hopefully we are very close to being able to realize this, is we want to have two microcavities, as in this picture, each existing in a different arm of an optical interferometer. So basically, we want to send from the left a single photon, split it at a beam splitter, and then get it to simultaneously interact with each of these cavities. And if inside these cavities you really put something like a virus or a bacterium or whatever you can really manage in this context, you could get one of those interesting states where one of these systems is excited and the other one is not, and at the same time the opposite, where the other system is excited and the first one is not. And then the rest of this image shows you how you could maybe from the emission spectra of these two systems tell that they've really been in this kind of entangled state, excited ground plus ground excited. So these are the experiments that we're trying to move towards. And of course, we can actually, this slide only shows you that we can scale it up in principle arbitrarily, because we can engineer these microcavities. We can engineer an array of 10 by 10 or 20 by 20 optical microcavities very efficiently and very quickly. And ultimately, you could imagine that each of these cavities in the image could contain an organic molecule itself. And then maybe by some suitable coupling to light, we can make a huge quantum coherent entangled state of these systems. So that's kind of the long-term vision that we're trying to pursue. And finally, I don't want to bore you anymore with this. I want to conclude actually. The conclusion is simply that as far as we can tell, quantum physics applies at all levels. And it certainly applies to large objects to the extent that I was talking about. People interfere all sorts of organic and inorganic molecules. In this case, I was arguing that even a living system can be in an entangled state and can show consequences of quantum superpositions. General tests of quantumness of all of these coherencies, superpositions, entanglements, they've been well developed in many body physics. We know more or less how to implement them. And I think the message here is that they are also applicable to living systems. I'm citing at the end of this a couple of recent papers that I've written with my colleague Chiara Maletto on precisely this, how to use quantumness to test quantumness of other systems, if you like. I'm making this conclusion. I can probably make it strongly. I'm putting it a little bit more cautiously, that there is probably no complementarity between being alive and being entangled. You know, I think these old ideas of Bohr were extremely stimulating, and I think they enthused a whole generation of physicists to go into biology. I think they led Schrödinger to write the book What is Life and so on. But actually, I think currently we've moved on, and I think if anything, we're very confident that quantum mechanics will be seen in more and more macroscopic contexts. The final question, which I think is exciting to all of us, and especially to your workshop, is really, are these quantum effects somehow, have they evolved? I'm putting it in inverted commas. Are they really deliberately there? Has the system been improved through natural selection to really reach this level where quantum effects are deliberately there? Or is it just an accident of the right conditions, the right way in which we probe the system, interact with it, and so on? And I think this is possibly for me the most exciting open question. Thank you very much.
Very, very interesting talk. I think we have questions. Yes, go ahead.
I guess I have a philosophical question, which is...
A microphone.
Can we do the microphone?
Oh, you need a microphone?
Probably not, but whatever. So at a philosophical level, I mean, what would have happened if you would have shown that, you know, a dead bacterium can be entangled and a live bacterium cannot be? We would have had to return to vitalism of the 19th century. I mean, I mean, the experiment is beautiful to show that, but I mean, it's almost hard to believe how you could have gotten a different answer. I mean,
you're absolutely right. I think we're talking about all of these chloroforms existing in the bacterium. And I doubt that they would couple any differently if the bacterium was dead to if the bacterium was alive. So you are absolutely right. This entanglement that we observe is most likely completely independent of whether the bacterium is living or dead. It's a very good point.
I had another question. So in your treatment, obviously there is no difference whether it's a bacteria or a whole sheep. You know, it could be
Absolutely.
goat, elephant, anything you wish. You know,
it would be hard to squeeze a sheep into a microcavity.
Well, you can make you can put it into a macro cavity. You can put it in a macro cavity. But I mean, so I mean, I really think that. My question is, obviously you treat these dipoles as independent, and if they were just independent dipoles, you get the result you get. But obviously the electronic states are many body systems there, so they're not the ones that you have in your Hamiltonian.
Yes, you're right. This is an extremely complex. I think we've made a slightly more detailed analysis in another paper, but these are very complex states. So basically what happens when the light comes in, you get this exciton being created. And I think excitons are roughly up to a nanosecond time scale, long-lived. Then, of course, you have this polariton state, which is this exciton, which is already the complex state you mentioned. This is now a joint state with a photon inside the cavity. And this usually lives up to, you know, picosecond time scales. So I think, and then on top of it, the cavity is leaky. It's not a high Q cavity. I think the lifetime of a photon is roughly 10 to 20 femtoseconds inside the cavity. So the Q, I think, is between 20 and 30, the Q factor. So in a sense, if you really want to describe it properly, you should take into account all of these things and treat it as an open quantum system. And actually, this is very, very, very hard to do this analysis exactly.
John.
Hi, Vladko. John McFadden here. So this might be a dumb question coming from a biologist, but how stable is the entanglement and why doesn't it decohere? And can you use this system to look at rates of decoherence and stability of quantum states inside living systems?
I think, yes, this relates to the comment. It's actually a very good point. It's not very long-lived. The entanglement, like I said, this polariton state exists up to maybe tens of picoseconds. So it's very short-lived. I would really be surprised if this so this is an entangled state between the dipoles inside the bacterium and the electromagnetic field outside of the bacterium inside the cavity. So like I said, this is a fun thing to study as a physicist, but I think I'd be very surprised if this had any biological relevance. It would be very interesting. Possibly entanglement within the bacterium excitons is a different question. But I think this is all about entanglement between the bacterium and the electromagnetic field. And I think it would be really...
But surely that will be probing the level of... I mean, if if the system, the dipoles inside the bacterium are entangled with the photons, then decoherence going on inside the bacterial cell will break that entanglement, won't it?
Yes, you're absolutely right.
It can be a probe of how much decoherence there is in different systems, and asking the question, is the system tuned to avoid decoherence, for example?
Yes, this is a very exciting question. I don't think this experiment is really sensitive or refined enough to say this. So certainly these excitons live much longer than the polaritons, if you like. So I think if you think of excitons as telling you that the dipoles inside the bacterium are entangled, then that actually exists orders of magnitude longer than the timescales of the experiment. But how to probe into that and see whether it's fine-tuned is a beautiful question.
Jennifer? Yeah.
Hi there, Jennifer Ogilvie from the University of Michigan.
Hi.
I'm just wondering, can you really say that you've entangled the bacterium with the cavity? Or, I mean, to me, this is part of the bacterium. This is the chlorosome.
Yes.
Yeah, that's my question. Can you, I mean...
It's a very good point. I think, again, it's... I think you have to work again within this model where you take a very simple view of the bacteria consisting of a large number of two-level quantum systems, if you like, just quantized dipoles, if you like, and you couple it to this mode of the quantum field, optical field, and under those circumstances, if you believe that this is the right model, then of course it tells you that the state that they exist in is an entangled state. But if you really want to be certain that they're entangled, you would have to do something that seems to me extremely hard experimentally, which is to try to separate the bacteria from the light field at some point, and then independently measure the dipoles in the bacteria and independently measure the light field to verify correlations. So, this would be going in the direction of Bell's inequalities. And in principle, you could do that if they're entangled, but I don't really see how to extend this experimentally. It would be extremely challenging. So I agree, the conclusion, you have to be careful. The conclusion is correct, given that we really trust that the Hamiltonian that I wrote down, or a related Hamiltonian, is the right way to describe this.
So let me follow up on that. In the measurements that you showed, there is no direct evidence of entanglement, right? I mean,
no direct evidence at all.
Right. It's just the splitting of the frequency and the
exactly the splitting of the. That's why I said probably the next level up would still not be entanglement, but would be to show that you can coherently address these two levels. So I think, and to me, this sounds very doable, actually. So basically tuning a laser directly on resonance to these two Rabi vacuum split levels and driving Rabi flopping between these two. And I think that would already tell you a bit more about how quantum they are.
Right. Yeah. Anyway, I hope you will stay on. We've done some experiments on green fluorescent proteins that are not cavity-based, but traveling wave, where we have directly measured entanglement out of GFP molecules.
That sounds very exciting.
Yeah, thanks.
If I remember correctly, these are the bacteria live by receiving one photon every 24 hours coming from thermal
yes.
winds. So is there a specific reason selecting this system?
Yes, I think these are the ones that are required. I'm not sure if it's one photon. I think it's a bit more than that, but certainly very few photons. I'm trying to remember how much. There was a very funny quote. I think that it would take this bacterium longer than the age of the universe to synthesize the equivalent of a Mars bar, chocolate Mars bar worth of energy. So they get very, very few photons per day. And basically that's one of the kind of circumstantial bits of evidence that they are extremely efficient at catching them and processing them. I think these ones came from New Zealand, if I remember from Dave. They are anaerobic. They're very robust. I think he kept them in the fridge. Then he took them out, experimented, and they lasted for a long period of time during this. I don't think there was anything special on the biological side as far as handling and preparation.
So let me ask another question. I know that there is in the literature somebody's made a laser out of a living bacteria.
Right.
So in some sense, that's also reminiscent of this kind of experiment.
Yes. I think when we use organic molecules now at this level of quantum optics, then it's exactly used for single photon generation. And this, of course, is a big deal in quantum technologies because quantum cryptography somehow requires you to be confident that you have a single photon with a high fidelity. So you're right. You could think of them as mini lasers of sorts. And it's actually surprising that some of these organic molecules end up giving you a much higher yield than the artificial quantum dots. So I think it's possible to think of them in this way as well.
Right. Yeah. So I think I completely agree on that point, and some of the experiments that my group has done sort of point in that direction that you can create, for example, entanglement out of biological systems.
Yes.
Not to say that implies that the biological system internally is entangled, but certainly
yes.
You can use biological systems to create entanglement of the optical radiation that they emit.
Yes. Yes.
And this living bacteria laser that I mentioned also was a a living cell that's held in a cavity, and the cavity is pumped at the right wavelength where the GFP in that absorbs and then emits, and the emission is like a laser beam.
Yes.
So
I'd be very interested to read about it more.
Yeah, I can send you a link. I think it's been
great.
a few years. Yeah. Any other questions? I guess we move on to this. Yeah, so I guess the next we have 20-minute discussion, and you have to chair.
I'm not sure what I'm supposed to do in this discussion, but Vlatko is offline. Huh? Yeah. Yes.
Vlatko, can you hear us?
Yeah, I can hear you. Can you hear me?
Oh, okay.
Yes. Yeah. I can see you.
Okay, since
We're working on that.
Okay.
We'll work on that.
All right.
Rodrigo, you can see him? There we go. Good.
Okay, I think it's a good opportunity since Vlatko is around to ask him, maybe start the discussion by asking him what or how he sees quantum biology moving on in the next 10 years. Just educated guesses and.
Yes, I think that it's a very exciting field, and I feel that going down to the level of single molecules seems to me the most exciting aspect. I have a feeling we have enough. We certainly have enough evidence that there are genuine quantum effects in biological systems. Again, like I said, it's sometimes hard to tell whether this is telling us that these systems are capable of behaving quantumly when we probe them in the right way. And I think you will no doubt be aware of these big discussions regarding the FMO experiments, whether they are quantum because they're driven by coherent laser light and so on. I think there are lots of these discussions. Can an incoherent source like the sun, for instance, would it achieve the same thing, and so on. So I think there are lots of these interesting questions that probably would be easier to play with at the level of single molecules. But I think it's clear that we've observed quantum effects there. I think it probably is clear that quantum effects exist even under these incoherent circumstances, or at least to me, I think this is probably not an important aspect there. But then what I mentioned is, and I have no idea how this would be addressed, but I think the question of whether nature has really found some kind of soft spot. I think Seth Lloyd calls it the Goldilocks principle. That all of these frequencies, the coherent resonant transport frequency, the incoherent phononic vibrations, the coupling, they all kind of have evolved to be in the right regime to optimize energy or electron transport or whatever else is transported in these systems. I think to me this is a big question, and how far we can go with the biological handling of these systems. Can we genetically modify them to change geometries and then to test them to see whether quantum effects are still there, and so on. I think this is how I see the next 5 to 10 years.
And if you had to bet money, and since there are some talks coming up, do you think brain has anything to do with quantum computation?
Okay, interesting idea. I think again the difficulty with the brain, of course, is that we don't really understand this even classically speaking. So I think people still argue about what kind of logic of computation does the brain represent, if you like. It's clearly not the usual von Neumann architecture where you have a central processing unit and so on. So the brain is obviously extremely interconnected. And I think lots of these features seem to me completely classical. They're at the time scales where it's very unlikely that quantum mechanics will have any effects. Now, having said that, it's not excluded at all that quantum effects which take place at very short time scales and very small distances, they could still be amplified ultimately to be relevant for our thinking processes and so on. So I would say this is wide open, but so far we certainly have absolutely no evidence that anything like that is relevant. What I would like, to link it to the first thing that I said, I'd like to, I know that there are these speculations about microtubules being relevant for consciousness and so on. I think, again, as a physicist, if you could just isolate individual microtubules, and I think you can, and then probe them in the way that we've probed other biological molecules to first see whether they even can support coherent transport. This, to me, would be the first step to make. And then, of course, probably under some circumstances, you will realize that they can do that. But of course, then we will have to ask a very similar question. Is this quantumness really relevant for anything or not? So I think we need to make exactly the same steps that we've been making with other quantum biological systems.
I think this is on? Yes. I think we are dancing on the edges. The question is, which are the things that we know quantum effects import, and which ones we think that we should speculate about it? Dave made the quote from John that quantum mechanics is essential but trivial. I think it's true that if we take out the chemistry, there's lots of chemistry taking place, but let's forget about the chemistry for the time being. We all accept enzymes and chemistry takes place on the body. I think there is some understanding that basically you have electron tunneling and you have proton tunneling, and these effects are quantum mechanical, and they are important to be on this way. There's a little bit of more question about vision, but there is some belief that it should go there. The idea of energy transfer by solitons was very famous in the past and got completely debunked by saying it's not robust, it cannot be done. The word coherence has been loosely used around here. Maybe our chair was the guy was a little bit more formal on the definition, but everybody else has used it at their convenience. And sometimes I get confused. But these are the things we know that quantum mechanics is important. In biology, I don't have any other thing that's convincing until now. Maybe someone can convince me. But should we go beyond what I just state as possible effects that we believe that quantum mechanics take place? Or maybe I left out some sensoring systems. We have a little bit of magnetic sensory and other kinds of sensory. Vision is one of them, so I wouldn't go beyond just vision. But other than that, do we have some evidence? I think we're going to listen to talks on brain, we're going to listen to talks on that. We talk about this coherence communication, but do we have some evidence for that, or that's just wishful thinking? And I would like to get a feeling from the audience here about those topics.
Yeah, I mean, I can make maybe quantum yogurt. I mean, if we have quantum bacteria, we can make quantum yogurt. I don't know. I think that we have delved now really in the esoteric, maybe not even science, I would say. I think, so my personal opinion, and again, I should be very careful because I also fund this kind of research. But as Jose said, I think that we really need to think seriously where actually quantum mechanics matters. Because obviously, as Jose said, if you go to femtoseconds, everything is quantum mechanical. There is not, at very short time scales, very small distances, everything is quantum mechanical. So I really, if we can discuss maybe today a little bit more on which are actually, where function was selected evolutionary, because it was advantageous for the organism to actually use a quantum phenomena and had advantage compared to organisms that use some other phenomena for survival. Perhaps, as we said, magnetosensing and things like this are very, very interesting, very exciting. So.
Yeah. Hi, Vlatko. Judith Klinman here. How are you?
Hi.
Yeah. So the enzyme work, I just want to comment on your last question. Are quantum effects deliberate in living systems? And I think what we've learned looking at enzymes is that one of their modes of catalyzing reactions is to get the reacting molecules very close. And they do this through involvement of the large protein matrix. And because of that, we see tunneling very often, and we see it with properties that are different from bulk solution. But it's not really deliberate. The deliberateness is getting the enzymes to work very fast, and the mechanisms by which they do that lead to internuclear distances that are particularly conducive for tunneling. So I think, if I would say I would call that not deliberate, but a consequence of how biology has evolved. I don't know if anyone else wants to comment on that, but I think in that case, nothing is absolutely clear in this field, but it's pretty evident from the ways in which enzymes work. So.
Yes, I think if I can just quickly interject, I think you are absolutely right that even I've seen, for instance, studies where people look at the FMO complex and then they take different distributions of these sites where excitons can get created, and basically they search this space of all possible distributions of these sites in order to show that maybe the actual distribution is somehow closer to the optimum. But the fact that they are arranged in this way that you get an optimum transfer, and the fact that at that point it's also quantum mechanical, it's not clear whether the conclusion is that this is deliberately evolved to really harness quantum mechanics, or it's simply this geometry that allows you to have the most efficient transport. It's not clear how to separate the two.
Yeah, I think the question of whether something has evolved to do a particular task is extremely difficult to answer retrospectively in biology anyway. The only way that you can really do it is see whether something has a selective advantage in an evolutionary experiment. And those kind of things can be done. You can evolve organisms in vitro in the lab, and particularly with short-lived organisms like bacteria, you can evolve organisms to have a particular phenotype or to not have a particular phenotype. And then you can have a look to see if the factors that you're interested in, like coherence, like entanglement, do they get affected by your evolution? And that kind of technique has been used throughout biology, and we just haven't got there yet in terms of quantum biology to be able to do those kind of experiments. But that ultimately, you can't retrospectively remake organisms and allow them to tell you whether something has evolved for an advantage. You've got to break it in some way or improve it in some way, and then you can work out whether it's got an evolutionary advantage to the cell. And those kind of experiments we can do, but we haven't done many of them. I think Judith has done some with protein, with looking at mutations in proteins in enzymes in a directed way, but you can do it in undirected ways as well, which are very informative.
Melishana Urbana-Champagne. It appears that for matters such as coherence or entanglement, perhaps one has to ask if they relate to an evolutionary selection pressure in the form of growth efficiency of the bacterium itself. For a number of quantum mechanical properties, such as, let's say, extonic line shifts, or thermal broadening of line shapes, or extonic delocalization, if you fail to account for these in the operation of the system, you actually lose significant efficiency, which has, in fact, a multiplicative effect on the energy conversion efficiencies across toward the survival of the entire species. I'm not aware of cases where, let's say, an experiment has been performed where the coherence of a protein variant and a mutant that has a different coherence behavior has demonstrably have different growth efficiencies.
That's a question.
Are there experiments that actually...
You wanna comment on that? Okay.
I guess the answer is no.
Yeah. Can you hear us still?
I think it's breaking up, actually. I might have missed the last minute or two of the discussion.
Okay. Yeah. Well, if I may make a comment and ask you to also comment on that. One thing that became apparent is that everybody uses the word coherence in a different context. And before we address what Jose asked, is there evidence? One should come on board with how to quantify evidence in the first place. And even after many conferences in quantum effects in biology in Europe and elsewhere, for 10 years now, still people use coherence in a very personalized manner.
Yes. Yes, I agree with that. It's I think you have to be doubly careful, because on top of it, coherence is a concept from classical physics, of course. And when people talk about classical coherent light, there are spatial, temporal aspects of that and so on. And then on top of it, you have to discriminate whether you are at the level where now coherence really comes from quantum superpositions, and which degrees of freedom are you superposing and so on. So I think you're right. You have to be very careful when you claim that something is really quantum coherent, as opposed to just the way a classical wave would be. I agree.
I agree, but let's try to make the comment the way I think Creston was doing his quantum yogurt or wherever. So
Yes.
I don't know if the quantum yogurt is Bulgarian or Greek. We can talk about it also. But but the question is, there are a few phenomena were discussed here that was clearly quantum mechanical and clearly measured. And I think electron transfer and proton transfer effects like that have been clearly observed. We can, Judith has a lot of discussion between cause and consequence here. I want to stay out of it for a little bit. But most of biological systems, that's a word that's not mathematical, is used loosely, so I'm going to use loosely, tend to be robust. And every time people say something is robust, we have to be robust to something and we have to quantify. But just using this idea in general, a lot of the quantum effects that we discuss, like entanglement or things like that, they tend to be very non-robust. So the question is, if these things are going to exist in biology, how do you reconcile these things? How would you do selection for such phenomena? And I have difficulties seeing that. Right? Doesn't mean it doesn't happen. My ignorance doesn't mean it doesn't happen. But I think I would like to see a little bit more justification, particularly how a system would evolve. That would be clearly, entanglement would be clear, something we all agree that anyone's definition of coherence is a coherent effect. So there is some need. So things like that is where I get a little bit confused, because, not confused in a sense, because I haven't seen any evidence that puts it. So going out from this sort of molecular phenomena to some large mesoscopic scales and stuff like, things get much more complicated. And that's, I think, what you're starting to discuss, and I think it's a very interesting question, if we can at least say something out of this workshop.
Yes, I think you're right. If I may just briefly interject, you are actually completely right that we have to somehow discriminate timescales and the spatial domain beyond which quantum presence of things like quantum entanglement would really still exist, and it would be surprising, because if you talk about very short timescales, then of course everything is quantum at that level, and it's beyond doubt that this is quantum. So I think we are looking for effects which are kind of long enough, robust enough, that all of us get surprised. We say, oh, we didn't know it could exist for as long as a microsecond, spatially beyond a micron or things like that. And then I think you could maybe say that something deliberately evolved to protect this behavior. It's like a decoherence-free subspace, as we would call it in the field. So I wonder whether something like that would be the right way to think about it. I don't know.
I would say that, sorry.
Yeah, I'll just go quickly. Okay, so any isolated quantum system is quantum coherent. So what I think we're really talking about is isolation and decoherence. So when the isolated quantum system, which is coherent, couples to a larger bath or an environment, that causes entanglement with the bath, and you can call that decoherence if you want. So I think we really want to focus on the notion of isolation and protecting maybe decoherence-free subspaces, as you're saying. I think that would be.
Yes, and in these complex molecules, actually, it's even hard to separate this into the system and the bath, because we don't really know whether the bath itself is highly, non-Markovian and all sorts of other, long coherence, spatial coherence, in which case even the bath is part of the system.
Right.
So it's not even clear whether this is the right way to go. I agree.
But actually, I disagree, and I think you just disagreed too. I mean, separating
I think
a system.
the point is that the division, it's very important when you put the line somewhere. Certainly, there will be degrees of freedom which we cannot control, and you can call them the environment if you like. But actually, it's in a complex molecule. What is part of this environment is highly unclear to me, at least.
Right. I mean, that's why I want to make the same point, that you can separate always part of the system and call it a bath. That doesn't solve the problem. I mean, you can think of the whole universe as isolated, and then you say the whole universe is quantum, but that doesn't solve the problem, actually.
Yes. I mean, certainly at the level of the universe, it would be trivial. Although we don't know that, by the way, that's a big question in its own right. But yes, I understand what you're saying.
But following up on that, I wonder if thinking about the cell and what part of the cell you call the system and what part of the cell you call the bath, is that an interesting question in terms of quantum mechanics? If the information from a system, say a protein or a piece of DNA, has not leaked outside of the cell but is leaked into the environment of the cell, is the system quantum or is it classical? Because from an outside perspective, you're not entangled with the system, with the degrees of freedom of the system, but inside the cell, something is entangled with it that is, to a certain extent, separated from it. And I wondered is there something interesting in the borders between living systems and the organelles of living systems and the borders of the cell, and what you call the system and what you call the bath? Can it make a difference to how we think about things like coherence and entanglement and how long they survive within living systems? Is it at all interesting or not? I'm not sure. Or where we draw the border.
It sounds to me like it's a very interesting question, because to call something a traditional bath, probably what you want is that these degrees of freedom rapidly thermalize. So as soon as you get anything from the system to go into this, it basically becomes thermal. It loses any memory of the previous state, and therefore, you actually can somehow separate the two without making any mistakes. Now, where this borderline is, it could even be within this complex molecule, that the vibrations, maybe under some circumstances and for some processes, you can think of as a traditional bath, but maybe under some other circumstances they are not like that. So that's what I'm saying. Certainly at the level of the cell, I don't know. It's really complicated.
And there are many systems inside cells that are non-ergodic, that have memory, and looking at how those are behaving is telling us that actually not everything is escaping from the cell. Some of the information is retained by the cell and can be kept in the system. So I think it is an interesting question.
Yes, it's very interesting.
Other comments or points or?
I don't think we have addressed the topic of this conversation. That was the feasibility of quantum information science in biological systems. Anyone want to comment on that?
Well, quantum information has the word information in it, and generally we think of holding information locally. And if you have a system which holds information locally, maybe the direction of a spin, say of an electron spin or something, if there is a bath that entangles with, that information is very, very rapidly lost into the bath, and the bath thermalizes. And so I think in order to have quantum information being relevant in the context of biology, one needs very isolated systems, which that coupling time to the bath is macroscopic. And I'll mention one of those in my talk later.
Actually, let me again, maybe Vlatko can address this. I mean, so if you look at, for example, I mean, I'll go to very esoteric area. If you look at photons that are emitted by a black hole, in fact, these correlations are not lost. The information is not lost. In fact, this bath, you can say it's thermalizing, but actually there are correlations in the bath that keep the information. Is that true?
That's another good point, actually, and this is how Schrödinger, in fact, understood entanglement in his original papers in 1935. So you can look at the two systems locally and measure each of them only locally, and they could appear to be very mixed. In fact, they could give you what you just call the thermal spectrum. But in fact, if you look at them jointly, they could be in a highly entangled state. So you're absolutely right that it's not enough to probe the system individually, because that's not going to reveal anything about the correlations with something else. So the system could appear to be thermal, but in fact, if you enlarge, if you zoom out, it's part of a highly coherent two-mode entangled state. And that's actually how this Hawking radiation is understood. I mean, bear in mind that the whole, we don't have a theory of quantum gravity. We don't actually know whether that's the right description of anything. That's a semi-classical treatment. However, if it is like that, then it is exactly how you described it.
May I ask something like a cultural question? How does the rest of the quantum science community that you're in touch with perceive quantum biology? Is it with great skepticism, or is it with excitement, or both, or something else?
I don't know. There is a, yes, you're right. There is a bit of both. I mean, to me, when I started getting engaged, it felt very much like how quantum computation felt in the 90s. It had this buzz, and I thought it's asking very fundamental questions and so on. So I think, if anything, there is certainly much more excitement about it than skepticism. There are many people who voice skepticism, but I think they are along the lines of the questions that we've been asking now. Nothing outside of that really. So the main skepticism is has there really been enough evidence that this is really quantum? And if so, then so what? For biology and so on. But I think the general sentiment is excitement. I don't know if you heard that.
Oh, oh, myself definitely, but I don't know if everybody. Okay, so I guess we have to wrap the session up. Thank Vlatko.
Thank you very much. Thanks a lot for having me.